Verma ma'am, please unmute yourself. You are not audible.
Yes, she is doing. She is doing.
Okay.
No, no.
Verma madam, you are not audible. Will it be possible for you to switch over to some other system over there?
सर किसकी आवाज नहीं आ रही? आप अनम्यूट करो ना, वहां तो आप अनम्यूट करो ना, अनम्यूट यहाँ से आवाज क्यों नहीं आ रही? क्लाउड को आप बोलो, क्लाउड को इनवाइट करो, एक मिनट करो, आप उठा करो,
ज्वाइन नहीं किया है सर ने।
आवाज नहीं आ रही, आप इनवाइट करो।
सबको नमस्कार एक बार फिर से। हमारे बीच हमारे अतिरिक्त संचालक महोदय डॉक्टर सुरेश टी सिलावट सर जुड़ चुके हैं। ऑन बिहाफ ऑफ द कॉलेज आई रिक्वेस्ट सिलावट सर कि वो यहाँ अपना उद्बोधन दे दे और उनके आशीर्वचन से ही हमारा ये इस वेबिनार हेलो हेलो सर अभी जुड़ रहे हैं। एडिशनल डायरेक्टर सर इज जॉइनिंग अस विद इन फ्यू मोमेंट्स। प्लीज बेयर विथ अस। Hello, Ranjana Verma, madam. Can you hear me? You please start the webinar. As soon as sir joins us, I will inform you. इसकी आवाज
Am I audible? Thank you.
Yes, you are audible. Ranjana, you can start the program. Sir, AD sir will join us. I will inform you. Okay.
Okay. Good afternoon to all. I, Dr. Ranjana Verma, Assistant Professor of Zoology. Pleased to welcome you all. We are delighted and honored to host the international webinar on emerging trends in biotechnology. A quality initiative by Biotechnology Association, Department of Biotechnology, under the umbrella of IQAC and World Bank MPHE QIPS, Academic Excellence Initiative, Berulal Patidar Government PG College, Mahu. This international webinar is principally designed to focus on advances in sequencing and computational biology. In today's scenario, researchers are exploring new methods in bioinformatics that help us in analyzing sequence and alignment, genome assembly, drug design and discovery, expression, and many more. Without much delay, I would like to start the program with the welcome note. Our heartiest welcome to our Chief Patron, Dr. Suresh Silawat Sir, Additional Director, Department of Higher Education, Indore Division, and Principal, Government Holkar Science College, Indore. We also welcome a renowned and eminent speaker, Dr. Priya Nevgaonkar, Madam, Department of Bioinformatics, Biocenter, Haberland University of Würzburg, Germany. Patron, Dr. Shobha Jain, Madam, Principal of our college. I also welcome each and every delegate for being present here today. Welcome to one and all. Now, I invite our Chief Patron, Dr. Suresh Silawat Sir, Additional Director, Department of Higher Education, Indore Division, and Principal, Government Holkar Science College, Indore, to bestow us with his valuable words. Dr. Suresh Silawat Sir. Over to you, sir.
सिलावट सर, आपका बहुत बहुत स्वागत है आज के इस अंतर्राष्ट्रीय वेबिनार में। हम सब इंतजार कर रहे हैं आपके आशीर्वचन का। सर, इस वेबिनार की शुरुआत आपके कर कमलों से हो। प्लेटफॉर्म सर आपकी ओर है। आदरणीय सिलावट सर। आदरणीय सर, अनम्यूट करना पड़ेगा माइक।
अनम्यूट कर लीजिए, माइक
कर दिया है। नमस्कार, स्वाता गुप्ता, आवाज आ गई?
जी सर, आ गई है सर आवाज आपकी।
आज भैरूलाल पाटीदार शासकीय स्नातकोत्तर महाविद्यालय मऊ द्वारा एक दिवसीय अंतरराष्ट्रीय वेबिनार इमेजिंग ट्रेंड इन बायोटेक्नोलॉजी अंतरराष्ट्रीय वेबिनार का आयोजन किया गया, जिसमें मुख्य वक्ता डॉक्टर प्रिया नेवरगांवकर, प्राध्यापक बायोइंफॉर्मेटिक्स, बायो सेंटर, यूनिवर्सिटी ऑफ वुर्जबर्ग, जर्मनी, एवं डॉक्टर राधा रानी, सहायक प्राध्यापक बायोटेक्नोलॉजी विभाग, मोती नेहरू तकनीकी संस्थान, प्रयागराज, इलाहाबाद, उनका मैं हार्दिक स्वागत करता हूं और अभिनंदन करता हूं। और आज के इस वेबिनार के लिए मऊ कॉलेज की प्रचार डॉक्टर शोभा जैन, आयोजन समिति की स्वाता गुप्ता को बधाई देता हूं एवं उसके लिए राजेंद्र कोचले, सुशीला श्रीवास्तव, मदन भास्कर, अनिता सोलंकी, मोनिका घोटे, मुन्ना लाल उपाध्याय, संजय राज सोनी, अनिता बातम,
इन सबको इस कार्यक्रम के सफल आयोजन के लिए बहुत बहुत बधाई। धन्यवाद। जय हिंद।
बहुत बहुत धन्यवाद सर।
Thank
और
you,
इसके
sir.
साथ ही ये मंच डॉक्टर रंजना वर्मा की ओर है।
Thank you,
Thank
sir.
you, sir, for your wishes. We are grateful for your valuable time you took to share your thoughts. Thank you so much, sir. Now, I invite Dr. Shobha Jain, Madam, Principal, Bherulal Patidar, Government PG College, Mahu, to blaze us with her motivational words. Shobha Jain, Madam.
मैम वॉइस अनम्यूट कर दीजिएगा।
सभी को नमस्कार। महाविद्यालय के बायोटेक्नोलॉजी विभाग द्वारा आयोजित इस एक दिवसीय अंतर्राष्ट्रीय संगोष्ठी में आप सभी का हार्दिक अभिनंदन है। सर्वप्रथम अभिनंदन है उच्च शिक्षा विभाग मध्य प्रदेश के जाने माने हस्ताक्षर आदरणीय अतिरिक्त संचालक महोदय सिलावट सर का, जो इंदौर के सुप्रसिद्ध महाविद्यालय के प्राचार्य का भी दायित्व निर्वहन कर रहे हैं। मैं हृदय से उनका अभिनंदन करती हूँ, क्योंकि उनका स्नेह, उनका सानिध्य और उनका मार्गदर्शन सदैव महाविद्यालय को प्राप्त होता रहता है। मैं हृदय से अभिनंदन करती हूँ आज के इस संगोष्ठी में विषय विशेषज्ञ के रूप में यहाँ वेबिनार में सम्मिलित डॉक्टर प्रिया नेउरगांवकर, जो कि जर्मनी के सुप्रसिद्ध यूनिवर्सिटी में बायोटेक्नोलॉजी डिपार्टमेंट की विशेष रूप से बायोइंफॉर्मेटिक्स बायो सेंटर एम हबलैंड की प्रोफेसर हैं। मैं मैडम आपका बहुत बहुत स्वागत करती हूँ। साथ ही स्वागत है डॉक्टर राधा रानी का, जो इलाहाबाद से हमारे साथ जुड़ी है और मोतीलाल नेहरू राष्ट्रीय तकनीकी संस्थान में बायोटेक्नोलॉजी डिपार्टमेंट में पदस्थ हैं। ये दोनों ही विषय विशेषज्ञ महिला सशक्तिकरण की पर्याय भी है। और आज का जो विषय है, जो कि बदलते परिदृश्य में और वर्तमान भारत की महत्वाकांक्षी योजना आत्मनिर्भर भारत, उसकी ओर लगातार भारत को अग्रसर करने के लिए जो ये विषय है, जैव प्रौद्योगिकी की उभरती प्रवृत्तियां, पर्यावरण, कृषि, स्वास्थ्य, चिकित्सा, पशुपालन, ये सारी विविध क्षेत्रों में अपनी एक विशेष तकनीक के माध्यम से राष्ट्र को एक आगे बढ़ाने वाली जो ये धाराएं है, बायोटेक्नोलॉजी की जो तकनीक है, वो निश्चित रूप से आज के विषय के प्रमुख आधार हैं। और हमारी विषय विशेषज्ञाएं जो है उस पर प्रकाश डालेंगे, जिससे हम सभी लाभान्वित होंगे। और न केवल हम लाभान्वित होंगे, राष्ट्र को, भारत को आत्मनिर्भर बनाने की दिशा में भी ये सशक्त कदम होगा। मैं महाविद्यालय परिवार की ओर से आप दोनों ही विदुषियों का हृदय से अभिनंदन करती हूँ। मैं अभिनंदन करती हूँ आज की इस अंतर्राष्ट्रीय संगोष्ठी में जुड़े समस्त सहभागीगण का, जिन्होंने सहभागिता करके इस संगोष्ठी को सार्थकता प्रदान की है। और मैं विशेष रूप से बधाई देती हूँ महाविद्यालय के बायोटेक्नोलॉजी डिपार्टमेंट के जो समन्वयक हैं, आज के वेबिनार के भी जो समन्वयक और संयोजक हैं, डॉक्टर राजेंद्र कोचले सर, जो सह समन्वयक हैं डॉक्टर सुशीला श्रीवास्तव मैडम, कन्वीनर हैं डॉक्टर मदन वास्केल सर, को कन्वीनर इनके साथ हैं डॉक्टर मोनिका धोटे, और वेबिनार का संचालन कर रही डॉक्टर रंजना वर्मा मैडम, विभाग की सक्रिय प्राध्यापिकागण डॉक्टर सुनीता साकले मैडम, प्रिया जायसवाल मैडम, रुचिता सिंह मैडम, मैं सभी को विशेष रूप से बधाई देना चाहती हूँ कि बहुत ही प्रासंगिक विषय पर उन्होंने इस संगोष्ठी का आयोजन किया है। और महाविद्यालय की आईपीएससी टीम, विश्व बैंक की नोडल अधिकारी डॉक्टर स्वागता गुप्ता मैडम, आयोजन समिति के समस्त सदस्य और महाविद्यालय परिवार सभी इसके लिए बधाई का पात्र है। और मैं पुनः बायोटेक्नोलॉजी डिपार्टमेंट को बहुत बहुत शुभकामनाओं के साथ बधाई देना चाहती हूँ और विश्वास है कि निश्चित रूप से ये वेबिनार न केवल शैक्षणिक गुणवत्ता में वृद्धि करेगा, राष्ट्र को रोजगार के क्षेत्र में आत्मनिर्भर बनाने के क्षेत्र में भी एक मील का पत्थर साबित होगा। बहुत बहुत पुनः धन्यवाद, अभिनंदन, धन्यवाद।
Thank you so much, madam, for your kind words. You have always been our source of motivation. Now, I invite Dr. Rajendra Kochle sir, Head of the Department, Biotechnology, Coordinator and Convener of this international webinar, to introduce our speaker. Over to you, Rajendra Kochle sir.
Thank you so much, ma'am. Am I audible? Respected Dr. Suresh Lawat sir, Additional Director, Department of Higher Education, Indore Division, and Principal, Government Holka Science College, Indore. Respected Dr. Shobha Jain Madam, Principal, Barulal Patidar, Government PG College, Indore. I want to introduce our advisory committee. First, external advisory committee member is respected Dr. N.K. Dhakad sir, ex-Vice Chancellor, retired DAVV, Indore. Our next advisory committee member is respected Dr. Asha Jwarkar Madam, retired scientist and head from CSIR, National Environmental Engineering Research Institute, NIRI, Nagpur. Our next advisory committee member is respected Dr. Abhay Pulke sir, Senior Scientist, CSIR, National Institute of Oceanography, NIO, Mumbai. Our next advisory committee member is respected Dr. Anil Kumar sir, retired professor and head School of Biotechnology, DAVV, Indore. Our next advisory committee member is respected Dr. MMP Srivastav sir, retired professor and head Government Holkar Science College, Indore. Our next advisory committee member is Dr. Sweta Kalbe Kulkarni Madam, Research Officer, University of Saskatchewan, Canada. And respected all internal advisory committee member of Barulal Patidar Government PG College, Mahu. Now, I start today's session. We all are living in technology time. During last 1 year, whole world is facing and fighting with pandemic situation. In spite of advances in science and technology, a single virus bound us to work at online platform. Globally, scientists are trying to find a solution for this problem in such extreme time. Bioinformatics is one of reliable tool which help us to understand, deliver solution for this. Our today's speaker lights us with her experience to power of bioinformatics. Now, I introduce our first session speaker. Our today's speaker, Dr. Priya Neurgaonkar Madam, is young, dynamic researcher from Department of Bioinformatics Biocenter and Hubland University of Würzburg, Germany. Her area of research is functional genomics and system biology. 2014 to 2017, she worked as project assistant at CSIR, National Chemical Laboratories, NCL, Pune with NCIM Group. There, she was engaged in project bacterial transcription sequencing and gene expression analysis, genomic mining to uncover the biosystematic potential of novel marine bacteria project, and in upgradation of NCIM activity. Then she shifted to Germany and joined Thomas Dandeker Group for doctorate degree. There, she is associated with Bioinformatics Group and doing excellent work on data sequencing and data mining. She is having wide experience of practical hands on training machine learning, bioinformatics, and molecular biology. She is well skilled with computer language like Python R, MATLAB, and Linux etc. She is well expert in NGS data analysis and phylogeny data mining large scale. During her tenure, NCL 2016 Pune, she was winner of Burgess Manual of Scientist Bacteriology T-shirt design contest received from Professor Dr. William B. Whitman, Director of the Editorial Office for Burgess Manual Trust. She was also winner of workshop on NGS data analysis organized by RASA, Life Science Information and Gene Path DX India in 2014. Her recent international publication on predicted MSC second epitome presentation of SARS-CoV-2 spike protein correlated to the case fatality ratio of COVID-19 in different countries will help us to find solution in scenario of COVID. Now, I invite Dr. Priya Neurgaonkar, Madam, to deliver her lecture. Over to you, Dr. Priya, Madam.
Good afternoon, everyone. Can you hear me?
Yes, ma'am. You're audible, ma'am.
Okay. Thank you so much for inviting me here at Bairulal Patidar Government PG College. And thank you, Rajendra sir, for introducing me. And I would like to first thank our chief person, Dr. Suresh Silawat, for inviting me. Also, I would like to thank principal of this college, Dr. Shobha Jain. Also, I would like to thank Dr. Rajendra Kochle sir, HOD Biotechnology, Swagata Gupta madam and Dr. Verma madam for having me, and also Shekhar sir for technical support, and also external committee members. Last but not the least, Dr. Monica Dhote for allowing and giving me chance to speak here. So now I will start sharing my screen, and we can start with my talk. Okay, so it's my pleasure to introduce you to my city. Right now I'm living in Duisburg, which is central part of Germany. As you can see, it is a beautiful city. And this is my university where I work. And here we have various departments such as microbiology, biotechnology, bioinformatics. And I'm here in this department where we study functional genomics and systems biology. So today's topic is all about technology, leap towards technology, and how we can use these bioinformatic tools to tackle the pandemic. I have kept my presentation as simple as possible, and if somebody has questions, you can raise your hand and we can discuss it maybe afterwards, or you can also ask me questions in between the seminar also. Something went wrong.
19 तारीख लास्ट डेट करवाओ मैंने एक्टिव कराओ कोई दिक्कत नहीं है जरूरत कराओ मैं मिलिट्री में कराओ.
Okay, first of all, I will give you overview about my topic. So I will introduce you to the central dogma of microomics world, and then we can talk about sequencing technologies which are coming into the market and how this is re-evolving with the world. What is bioinformatics? What are different applications, databases, sequence alignment, and then we will talk about what are genomics, transcriptome technologies, and proteome analysis.
मतलब और ये बात हम उनको भी बोल चुके हैं और
In the last part,
ये बात वो भी समझते हैं हालांकि, पर ये सारा बढ़ रहा, अब ये सप्लीमेंट्री एग्जाम अरेंज कर रहे हैं तो
in the
ऐसा ना हो
last
कि वो
एक्सक्यूज मी जैन मैडम, आपकी माइक म्यूट कर दीजिए.
असाइनमेंट जमा करने के बाद रिजल्ट हो.
yeah, so thank you. In the last part, we can talk about what is the role of bioinformatics in COVID pandemic. So here I have some basic notes. Like, as you can see, this is the bacterial cell, and in this we have DNA. And when we come to any microbiology or biotechnology class, our teachers teach us that this bacterial cell grows inside your flask or test tube when you give optimum pH, optimum salt concentration, temperature, etc. And if you don't give it, it will not grow, or it will grow in less concentration. And who determines this is the DNA, right? Everybody will agree with me. And if we concentrate right now, if we are talking about, suppose, about motility, we will concentrate on the flagellar side of this bacteria. And if this DNA allows this bacteria to synthesize the proteins which are necessary for this bacteria to move, then it will start producing these proteins and this bacteria will move. So everything is dependent on DNA. It gives you or bacterial cell to synthesize some proteins and function inside the cell. So what is the DNA made up of? It is made up of 4 bases: adenine, thymine, guanine, and cytosine. And if we join these bases in certain fashion, it will give rise to some gene. And these genes are very important. And if we consider one human being, there are 40,000 genes which are coding for different information, how we function, how we develop our brains, or how we test some food, or how we talk, or how we grow. So these are very important. And if we uncover this, if we want to uncover these genes, we need to concentrate on our chromosomes. That means we have to study nucleic acids. That turns out to be DNA, and then it gives rise to genes. Now, not only DNA, but if we go further, there is RNA. More functional is mRNA, which gives rise to proteins with the help of ribosomes. And then this bacteria will start moving, in this case, if we concentrate on flagellar motility. This is, in simple words, it's like preparing biryani. If you want to prepare biryani, you need to have some recipe. And if you don't have recipe, if you teach some foreigner, if I give some foreigner, this is the biryani, and if you have to cook it, if you don't provide this recipe, it will not he will or she will not be able to prepare biryani unless she has this recipe and some sort of instructions and some material. So if we consider proteins as biryani, we see that these are the functional molecules. And these are very important for growth of organism. And there are set of proteins which are acting together to grow this bacteria. Now, if you ask cell question how you make a set of proteins inside the bacterial cell, it will tell you that I have written this program in the form of genes, and these instructions on the genes are written in ATGC. Why very important part in sequencing technology to understand how these proteins and DNA or RNA works, because nowadays you see that technology has changed. Like we used to study DNA, then proteins, and now everybody is talking about RNA vaccines. So we have to understand each and every step in central dogma. Now, if we think of what is the power of this DNA or protein, we will understand that if we consider this one human being, and if we take out some cells, and if we measure one DNA, length of one DNA out of that cell, it will measure up to 2 meters. And if we take out all the cells, that means there are 100 crores of cells inside our body, it will be 2 crore kilometers, which is actually distance from Earth to the Mars or Moon. So this is very huge amount of information in our DNA or in RNA and proteins. So to understand or to analyze this information, we need to have some tools, right? So bioinformatics is one of the tool to speed up this process and to understand and visualize this sort of complex information. Now, to make it interactive, I have this picture who can tell me, like, I saw some comments like good morning, good afternoon and everything, but who can tell me what is this? I believe some students of bachelor's or master's will know this, right? No? Look at the numbers. Okay, no problem. I can't see it in chat box, so I will tell you what is this. So you see 1 to 22 numbers. Okay, Rani Puranik has raised some hand, and you can type Rani into your chat box, or you can tell me.
Hello, ma'am. Am I audible?
Yeah.
Good afternoon to all. Ma'am, जो आपने पिक्चर शो करी है, I think it is sequencing of nucleotides.
Yes, but it is very specific, like which sequence has been sequenced using this, or what is this, like sequencing of what?
Nucleotides.
But which organism, can you guess?
No, I can't.
Okay, so I will give you a hint. So there are 22 chromosomes, and there are X chromosomes and Y chromosomes.
Yes, ma'am. That is autosomes, I think.
So these are the human genome.
Yes, map बोल सकते हैं. We can say human map, genome map.
Yes, exactly. So this is, thank you, ma'am. You're right. This is the human genome sequenced in 2003. So this project was started in 1990 and it was completed in 2003. As you can see, if you open one of these books, you will find that there is information about each and every chromosome inside these books. And if you want to try out to read this information, I will give you some snapshot or one snapshot. So you can't read this information if you say that you have book in your hand, and if you try to read some Harry Potter book, you can't read like this book in your computer or in your iPad also. So you need some tools to understand or to convert this information into the language which we understand. So before going to that, I would like to give you some story or tell you some story how this DNA, or we should be grateful to all these discoverers of DNA. So we know that this is the DNA and double helix structure and semi-conservative model, everything we know now. And because of that, we could do our research. So these are the two scientists, Watson and Crick, who discovered this model. And now they look like this. And Crick Institute is also there, and Watson is also working on various projects right now, and he is very active on TED Talk. If you want to listen to him, you can listen on YouTube, and very brilliant scientist, I would say, because he completed his PhD at the age of 22, and then he joined institute in US, and then they tried to work together. But he was a physicist. He wanted to study the structure of DNA. But what happened is that both of them were working together, but they could not find what is the structure of DNA, how we can unravel this. But after some time, they found it, and how that I will tell you. But because of that, they received Nobel Prize in medicine with along with Wilkins. Now, as Shobha Jain madam said that this is the power of women's, so there is lady behind this, and there is a conspiracy theory that Rosalind Franklin was one of the was having major role in discovery of DNA. She was also working in the institute and other institute, and she was working with the help of X-ray crystallography, and she found this photo 51, which is actually the X-ray crystal structure of this DNA helix. And this Wilkins was working with her, and he took some of the photographs from her lab, and he gave it to Watson and Crick. And what happened is, before she could publish it, because that was very complicated data. So अगर आपने सोचा होता कि आप अगर इस सीक्वेंस को और इस स्ट्रक्चर को एनालाइज करना चाहते तो it would have taken 1 year, and she took some time. Before that, they had taken all her research and tried to predict some structures and already published it, and the credit was not given to Rosalind Franklin, and Nobel Prize was not given to her because she had died because of cancer before that. But we should thank these scientists for giving us structure of DNA, so that we can design these vaccines, all these technologies which are available today. Now, coming to central dogma of biology, there is DNA, as I said, there is mRNA by the process of transcription, and then there is a protein. And if I study DNA, I study ATGC, and we have genomics. If we study mRNA, we have transcriptomics. If we study protein, we have proteomics. And there are various techniques by which we can study this. So this is known as omics world. And if we consider only genome, we study genotype. Like Mendel has given us recessive and dominant gene theory. Likewise, we study genotype. And if we study all these three parts, we focus on phenotype, that is morphology, functional part, how the pathways are behaving, or in ecology, how mutualism or altruism kind of phenomena behave. And this is very important when we speak of transcriptomics and genomics. So what is transcriptome and what is genome? So if we take entire genome, and if we put or if we extract DNA and if we sequence it and we understand information out of it, that is the sequence of the organism, and that is known as genomics. And transcriptome every RNA molecule produced out of DNA. And if we take expression levels at each level, at each point of time, then that would be the transcriptomics. And why RNA is important? Because you have entire set of DNA, but if we give some certain temperature, certain type of genes would be expressed in that organism. Not all of them would be expressed at the same time. So if we have some sort of hint that these are very important for this temperature, we would understand what are the proteins which are required for this by this organism to survive at this temperature. And likewise, we can study different characters or parameters. I will show you next some slides in that you will understand more. So here I have DNA, and if we take this DNA, there you can see there are and then we have RNA. So this is the DNA, and if we take coding parts of that DNA, it will code for short RNAs, non-coding RNAs, miRNAs, so microRNAs, short RNAs, and antisense. So all of them are working together to form this gene. And if we target particularly this gene, it is a huge set of instruction. We can't understand it. And we can't also study this RNA with this RNA, right? So we have to have some tool. If I give you these type of RNAs, you would be like, madam, please मुझे ये नहीं चाहिए. मैं अच्छा है, मैं दूसरा कुछ कर लेता हूँ और कर लेती हूँ क्योंकि I don't understand this information. So before that, information को बाहर निकालने के लिए we have to have some sequencing technologies, because if I say I have DNA, but in test tube, if I give you, you will not understand it. So for that, you need sequencing technologies. You must have studied it in your molecular biology paper, that there is a Sanger sequencing method, which is also known as chain termination method, which uses PCR fragments. And if you add your amplified DNA from your PCR tube, and if you provide some DNTPs and DDNTPs, with the help of chain termination method, it will give you the sequence of the DNA with the help of laser. And how this works is very well explained in some YouTube videos, as well as in Leninger, which is one of the good books for master's and bachelor program. Now, I would like to again thank one of the very important scientists, Sanger, who won Nobel Prize for discovering this Sanger sequencing method. With the help of that, we can sequence the DNA. And there is one of the research institute in UK which is focusing on sequencing technologies and understand how organisms work. And this is in UK. If you want to pursue your career, you are always welcome to go to Welcome Genome Campus. And there are some lacunas in this method, that it can't sequence, or it takes some time. So there is another method like Illumina sequencing. And this is very, very clumsy diagram, I would say, because you don't understand anything. So in simple words, this is a DNA fragment. And then you attach adapters to it. And then you create these kind of fragments. And then you add your, this is your original DNA, and then you adapters, and then you add simple nucleotides to it. And with the help of, again, fluorescent, this sequence can be understood by sequencing by synthesis method. And you get this kind of figure after sequencing. And each color belongs to one nucleic acid. And then you, with the help of softwares, you can understand this information. Now we are coming to, from your bacterial cell, we have extracted the DNA, and from DNA, with the help of sequencing, we now have this ATGC form of sequence. Now, as I told you in last few slides, if I give you book of ATGC, you will not understand what is this. You will say, I don't want to study this. So what is the next thing is, we want to have some softwares. So before going to that, I would like to say that why everybody is saying that bioinformatics is very important, or why this field is booming now, because there is new sequencing technology which involves RNA sequencing, and with the help of all these technologies, we can understand every organism like human, plants, mouse, or Drosophila, or any other. And why this has happened, like our mobile prices are going down, or and there are new versions of mobiles are coming. Samsung, we had Nokia in the first century, and then we had many Samsung, Apple, and everything. Same way in sequencing, there are different versions of technologies are coming, but the sequencing price is going down. So now we can have more of sequences out of the sequencing machine, but there are no there is no manpower available to study this, because they require certain skills to understand these type of information. So for that we need bioinformaticians. So how we can use these sequencing technologies? One example is you to get out of to get personalized medicines for the treatment of cancer. So as I said, in 1977, Sanger discovered this sequencing method. Then we had PCR. Then we had Human Genome Project. Until that, we are developing new methods. But we need to go further, and we have to have some personalized medicines, because if we provide every patient with chemotherapy or some sort of medicines which are available for cancer patient, it is not working. So sequencing technology could be one of the important medicine, I would say, to understand or to treat these cancer patients. And for this, after sequencing, we need bioinformatics. And for that, we need bioinformaticians. As I said, one of the latest technology is single cell RNA sequencing. In cancer, you have, so if I take example here, I have some tissues out of cancer cell. If we say that this is the cancer cell, and a red colored one is a cancer cell, and if we take out RNA and if we sequence each and every cell, that would be single cell RNA sequencing. And with the help of this this technique, we have clustered same cells into one bubble, right? And here you see that there are only 3, but if you have some cancer patients, they will have 4th bubble also. So that means that this sequencing technology and bioinformatics can give you clue about what are the disease cells which are associated with this type of disease condition. Now, I told you about all about sequencing. Now, what is bioinformatics? This is the conceptualizing biology in the molecules and applying informatics, and it includes CS, statistics, and all information on large scale. Who understood the definition? Nobody. You will say, "Arey bhai, aakhir kya kehna chahte ho," right? So, to give you a simple view, you can have sequencing data. You can involve computers, do some calculations, and you will have information. So it combines biology plus computers, some mathematics. Don't be scared of mathematics. You will understand when you enter into this field, and with the experience, you will be pro into this technology world. So very important is biology and computers. Now, what are the applications of this? You can manage your data. There is huge amount of data. So you can manage with the help of bioinformatics. You can develop new algorithms. We can analyze and interpret DNA. We can predict some drugs and discover some drugs. Also, we can predict some protein structures as well. So it is very important field, I would say. And then, but there are very few limitations, but they are very important. What are they? They need further research or experimental analysis, because you can't just predict it. You have to do it into the human beings, or you can do the next experiments into mouse or some model organisms. In case of plants, you have Arabidopsis. You need to have accuracy in these kind of methods. There are some errors involved and that can be fatal. So you have to be very precise. And in some algorithms, like in sequence alignment, you can have incorrect predictions. So this is very nice and glamorous technology, but it has some drawbacks. Now, a very important part of bioinformatics is databases. And what are they? These are computerized archives which store and organize information which we already have submitted to these databases, and we can easily retrieve those information. So and this can involve hardware and software. So what I mean by that, I will come to next slide and you will understand it. So you do experiments in the lab, you sequence the organism, you submit it to database, and then there are curators. They will curate the information. That means that they correct your information, and then it will be in the database available. And then reviewers will be the person who say that with the help of curators, they work together, and then the database can be created. Now, where does this data come from? With the help of experts, curators, and reviewers, there would be a data which would be available for you. You can also retrieve some data. How we can see into next slides. Now, if I give you some sequence, so these are very vague picture, right? There is Sachin Tendulkar, there is Sonia Gandhi, then there is Karishma Kapoor. Everybody is here, and you don't understand what I'm trying to say. So if we have protein sequences with DNA sequences and RNA sequences together, you won't understand. So for that, what the database do is they cluster or they group together information which looks similar. So these are the DNA sequences, these are the proteins, these are mRNA, and there are molecules. So biological databases can be divided into primary, secondary, and specialized databases. Now, what are primary databases? Gene bank is one of them. Protein database bank, and then there are many of them. So one by one, we can go to them. One of the mostly used databases is NCBI. If you go to Google right now and if you search for this, you will get most of the information is available here. So various databases are integrated into NCBI. As you can see here, if you want to study taxonomy, sequence analysis, you can do everything here. Same way, we have protein database, and you can predict the structure of protein here. And to understand protein data bank, there are various softwares available. You can search on internet. Like RasMol is there, PyMol is there to predict these structures. And to give you a glimpse of how the file of DNA looks is something like this. So you have a raw DNA sequence, which could be coding or it not be coding for something, but there is some information available, and you can study this information into the software, such as you can give this sequence here and you can get annotated part of gene into NCBI BLAST tool. Likewise, you can have protein structures with the help of 20 alphabets, because protein is made up of 20 letter alphabets, not ATGC, and this is also very complex information. Now there is another database which is EMBL, which is very important because they will provide you understanding of not only the sequence analysis or the BLAST, but you will understand what are the pathways which are governing all these processes, and you can visualize the pathways also. And I'm telling you all these databases so you can search them online and you can play around with them and come with the information which can help you into understanding of your biochemistry or molecular biology paper. So there are tools available, and there are resources which can provide you tutorials, and then there are various online platforms available to learn all these things. So you should take advantage of all these databases available online in this lockdown time, and play around with these tools which are very important in bioinformatics. Like biomodels is there, complex portal is there, where we can study models and pathways. Now, if we come to secondary databases, these are more precise if you want to have next sort of information apart from primary databases. These can give you more information or precise information. One of them is SwissProt. So if you have protein sequence, you can go to SwissProt or UniProt, and you can see whether this structure is already, or this sequence has been reviewed, or it has been studied yet or not, or you can go further and model this protein sequence into SwissProt and come up with the new idea that this protein, suppose we take spike protein from coronavirus, and if you discovered now and if you submit it here, and your publication would be ready. So you can study your sequence with the help of all these UniProt or SwissProt tools available. Then there are various other data sets available. There is plenty of information for protein also, like Panther is there, PFAM is there, because there are protein families. And if we consider, suppose, enzyme transferase, there are various residues are involved in binding of the molecule to certain type of molecule. And then there are active information, there are passive information, and then there are inhibitors. So to target all these molecules, we need all type of signature databases, and all this information is available online. And then there are specialized databases. If you want to study some type of worm which is available only in Madhya Pradesh, suppose, then you have certain databases which are very targeted to these kind of specialized information, and you go to them and you retrieve the information, and then you can study your organism. Likewise, here I'm showing you various databases. UniProt, Interest Gene ID, HGNC symbols are very important to study these gene IDs and do functional database analysis. And with the help of that, what you can do is predict some pathways. You can study gene expression analysis, and you come up with the disease terms and chemical terms, which can give you the entry towards discovering some new drug, or to predict which are the drugs we can which are very important in lab experiments, and then you do your lab experiments. So how this data cycle works is, you submit your, suppose now if you have DNA sequence, you submit it to the data bank, you can archive it, that means that will be stored. Then the database will take care of your information, and they will give you authorship. They will classify your sequence as DNA or RNA or protein. And afterwards, if you want to analyze it, or if I want to analyze it, I can access to that information here, and I can use various web tools, and I can come up with my idea. So how databases works, I have given you small a glimpse of it. This is very complex because there are many databases, there are very rich information available there, and you have to have various staff members allotted for each and every step here. So that's why you see some of the NCBI or EMBL platforms which require, or which have so much staff members involved to understand all these processes or procedures at each step. Now, the next part here in bioinformatics is sequence alignment. So if we take out the sequence, what we will do is first align the sequence. That means we have one sequence and we align with another sequence, and we see what are the similarities and what are the differences in the patterns or in the symbols which are there, and we come up with the information. And here I'm showing you one of the example. Like if you have retrieved, suppose all 4 sequences from NCBI, and here you can see there is VTPGIR. So this is the protein sequence, and there is repetition of this part here. So maybe this part is very important for binding to spike protein in case of coronavirus, and you can say that very important part is present in every SARS-CoV virus I have isolated from 4 patients. So it is providing you some information about coding sequences as well as non-coding part also. So alignment is key to finding important regions, as I said here. Determination of function, because if I take out only one part or one gene out of it, one protein from this, it will give me function. If I put it into some tool or software, it will give me function of without doing some experiments. It will uncover evolutionary forces. I'm talking about only the information available. If you want to have new information, you can do experiments and then you can come up with the new information available. Now, how the whole workflow looks is you get sequence, Sanger or Illumina sequencing, and then you do this sequence alignment. And with the help of sequence alignment, you can either go for diversity studies, or what are the relationships between these proteins you can study. Also, you can study taxonomic distribution, like if it belongs to some bacteria or virus or some human, and then you can understand what is the evolutionary or phylogenetic analysis of this bacteria can be done, and you can also discover some new molecules. So all these can be done in some of the softwares which are developed by various scientists all over the world, and one of them is Clustal Omega, and then there is Muscle. Everything is available online, or you can write me an email and I can give you information if you need for your experiments also. So now we are going into some of the complex things. So here I have DNA, and if I visualize all the information available online, I can visualize it into this kind of figure, which gives you idea about what are the Y coordinates from the human genome, what are the X chromosomes, what are the highlighted regions are important for. So you can see each and every chromosome is available here on this graph, and these are the important genes for that particular study, which was studied in 2017. So to create these kind of figures, you need DNA sequence available, and very important is bioinformatics softwares, because without that you can't convert this information into this kind of form. And this is very important when you want to study map together with the other information. As you can see here, there are 2 layers. So they have studied 2 human genomes together, and you can layer more human genomes, because nowadays sequencing has been cheap, and you can study patient-wise data here, and you can come up with the new ideas for drug discovery. As I promised you, we can study transcriptome here. So here is the example for it. You have control mouse, and then you have another mouse, and if you have 2 conditions and you come up with the 2 transcriptome, and you see here that there are 2 conditions on this graph or heat map. And you clearly see that there are some patterns which are repeated. Here you can see blue is there, then there is red, but there is no blue here again. So if we provide this condition, so there is some change. So you can say that the transcriptome is changed over here. And what are the genes which are changed? You can understand it by various Venn diagrams, and you can cluster them together. Whole thing is very complex. I know for the understanding point of view, here I'm not telling you to understand everything, but my point of view is to motivate you to further study all these terminologies, and you can do your PhDs or masters into these fields. So the important point here would be the third point is we have studied genomics. Now we came to transcriptomics, and the last point is proteomics. You can study protein with the help of electrophoresis, and then you see the protein fragments in MS analysis. And these MS analysis can be further analyzed by various bioinformatic tools, and you can come up with this nice diagram which tells you about how these molecules are interacting with another type of molecules. So these are, suppose we take example of RNA binding proteins, and if we want to study how actin binding filament are interacting with RNA binding, or what is the role of them, you can connect it through protein information available, and you can visualize it using various bioinformatic platforms available. Coming to last part of my talk is about understanding questions related to COVID-19. Last year we faced pandemic. Right now in Europe there is a lockdown, and we are very scared of lockdowns now. And as sir said, that this one small thing has changed everything nowadays. So we need to ask some questions to this virus so that we can understand its how it causes these infections, and how we can cure these patients, because sometimes it can be deadly. So there are now various data sets available online. You can also go, because all information is open and freely available, and you can also analyze the data available and interpret the results. How you can do it? Oh, there it is. Yeah. So here you can see that various affected regions. As you can see, here is India, and here I am in Germany, where is Europe is one of the affected area, and there is America. So various parts are affected, and you can also create these kind of diagrams in R softwares or in Python. And for that you need information, and there are various databases. As I told you, you can search COVID databases on Google and you will find information. Now if we consider transmission of corona virus in different species, you can see that not only coronavirus, SARS-CoV-19, but there are various viruses which are already being studied, and which are, suppose we take this dog, it has another type of influenza, and then there is a pig, and then there is a cat. Every coronavirus will have, or the influenza or this family will have similar structures, but they are functioning in different way. Because of that, they have various host ranges here. Their host is very important when we consider this spike proteins involved. And to understand these spike proteins, you need to sequence them, or you can precipitate them in the lab, and you can study the information available with the help of various molecular biology and bioinformatic techniques. Now, as I told you, you can get this type of sequence from NCBI, and if we take out all the available information, and if we plot phylogenetic tree, it would look like this. And you can say that my COVID-19 is very different from bat. Likewise, nowadays they are sequencing all the information available and coming up with the phylogenetic analysis. Now, here I'm showing you in last part how you can take out data from various patients, or for you it would be the database, these patients who are affected. And so this is the clinical sample 1 and this is the clinical sample 2, and we sequence it, and then there are viral dominant proteins, and in this sample you don't have Manmohan is raising hand. I don't know what. Yeah, you can ask.
Ma'am, please you continue the lecture.
Okay.
After this,
Okay.
we'll take over the questions.
Okay. Okay. So, as I said, if we take two individuals and there is affected sample, there is unaffected samples, and if you sequence it and then there is a dominant viral genome sequence available in the software, you can see various patterns, and then you see that there is no coronavirus here, and then there is a coronavirus dominant sequence here, and then you say that this patient has being infected with the COVID, or this is affected, this lady is not affected by the COVID. Likewise, there are various stories available on these platforms, and you can go through them, and they are actually accelerating the COVID research. Here there is open access data from INSDC database. Then there are drugs which are available in the market and which are very important in hijacking the cells, and how bioinformatics can develop new drugs can be understood using this type of stories here available on EMBL websites. And in this slide, I'm showing you how bioinformatics can be applied here. So virus, if we take example of COVID, this is the COVID virus, and then there are proteins from the human, and we can study these protein and virus host protein interactions with the help of this explorer, which is known as COVAX Coronavirus Explorer. And in this tool, we can also study protein-protein interactions. And then there is a drug-protein interaction also tool available in COVAX, and you can always go there, and this is very recently published by Oxford, and the information is very reliable and authentic from various type of patients available. And as you can see that there are various protein-protein interactions involved. It depends on the virus strain also, because you see that now there are various strains coming out of the coronavirus. Like there is London strain, and then there is South African strain. So their virus host protein interactions would be not 100% different, but there would be some difference in Indian origin Indian coronavirus now, and in Ireland coronavirus. So you can always study this online. And in my university, I'm targeting MHC2 epitopes to study these protein-protein interactions. And we say that MHC2 molecule is very important as far as these proteins are concerned, like nucleocapsid, spike proteins, and membrane proteins, and all other proteins. And we have also studied various patterns in different countries such as Germany, India, China, etc. And we wish to publish this paper soon. And yeah, we can come up with different ideas with the help of bioinformatics like this. So with this, I would like to acknowledge my university for allowing me to do my research for various funding agencies, and my fellowship giver DFG funding, which is German government, and my professor for supervising me for my studies. And I would like to thank you all for listening patiently. And I would like to thank Potidar College for giving me chance for sharing my research experience with you all. Thank you so much. And I'm ready to take questions. Thank you.
Okay. Thank you. Thank you so much, Priya Newgaukar, madam, for such a nice session, even brainstorming session. You have discussed a lot of things with us. Sequencing procedure, role of bioinformatics in sequencing procedure. Apart from this, you have discussed genome analysis in detail and basics of transcriptome analysis and proteomic analysis in developing procedure of COVID-19 vaccine. Thank you so much, madam. Now I invite Dr. Sunita Shakle, madam, for conduction of questionnaire session. Dr. Sunita Shakle, madam.
Thank you, Ranjana madam. Good afternoon, everyone. The clear and very effective way in which you presented your lecture help in no small way getting the webinar across. Your presentation skill were next to none. We can't thank you everyone for this informative knowledge you gave us. Millions saw the apple fall, but Newton's asked why. Many great discoveries came out of curiosity, so our curious participant want to ask some question. The first question I would like to ask you, is it mandatory to take COVID vaccine?
I could not hear you. Is it mandatory?
Mandatory to take COVID vaccine. COVID vaccine. Vaccine. Mandatory to take COVID vaccine.
What do you mean by tag? I did not get actually the question.
Vaccine.
Like you mean
okay,
tag?
Okay, now next question, madam. Is vaccine of COVID showing positive result? If you okay, if you saw why people have myth that it is having side effect on body.
Okay, so the vaccines are very important as far as COVID infection is concerned right now, because it is fatal sometimes. And there are vaccines developed all over the world, like AstraZeneca is there, and then there is Moderna, and from Germany there is Biotech. Now, to understand this vaccine part, sometimes it is very important for you to know these clinical trials are conducted on various patients all over the world. So the diversity changes from Asia to Europe and to the US. And the criteria in which this vaccine is studied in this clinical trials is very important. So that's why at the end of each trial, there is some committee members which sanction these vaccines to the next level. And the available vaccines are, I think it depends on also on the government policies, how they are tackling these kind of affairs in various countries. I think I tried to answer your question.
Okay, good. Thank you, madam. Over to you, Ranjana, madam.
Thank you, Priya, madam. Thank you, Shakla, madam. Now I invite Dr. Monica Dhote, madam, for conduction of second session. Over to you, Monica Dhote, madam.
Thank you, Ranjana, ma'am.
Monica, please unmute yourself.
Okay. I'm sorry. A very good afternoon to everyone. I am Dr. Monica Gote, faculty of Biotechnology.
Excuse me, ma'am, आपका मोबाइल थोड़ा सा
On behalf of Biotechnology Department, Bhairulal Patidar Government Mahu College, welcome all the respected members of panel, participants and friends in international webinar on emerging trends in biotechnology. Thank you, Priya ma'am, for such a wonderful talk. It was very nicely explained how information and demand of time. We explain it from the very basic to the application of bioinformatics. Dr. Priya has nicely explained how technology helps us to manage COVID and biological data and deliver solution for this deadly virus. Now, without taking much time, we proceed to our next session. During pandemic time, during last 1 year, coronavirus has teaches us power of microbes. But microbes are not always harmful. They are also beneficial to us. Many times, these beneficial microbes play dual role by utilizing waste and produce useful byproducts. They are used as food for energy production. MFCs, that is microbial fuel cells, is bioenergy production from microbes, is economic and reliable environmental technology in current time. Our next guest speaker, Dr. Radha, have wide experience of environmental pollution problems and their treatment technologies. Now I request Dr. Sushila Srivastava Madam to invite our second speaker, Dr. Radha. Dr. Sushila Srivastava Madam, it's now over to you.
Thank you, Dr. Monica. Good afternoon, distinguished members of the panel and all the concerned professionals gathered here. I am Dr. Sushila Srivastava, HOD Zoology.
मैम आपका मोबाइल थोड़ा सा
But area of specialization is environmental microbiology and biotechnology.
She has technical experience in bioremediation of soil contaminated with organic, inorganic pollutants, microbial fermentation. She did her doctorate from NCBI, Nagpur with CSIR fellowship. Her 18 papers have been published in international journals, 3 papers in conference proceedings. She authored a book in 2012 titled Bioremediation of Organophosphate Insecticides, released by Lambert Publication, Germany. Her chapters are present in 8 different books. She has also edited 4 papers and chapter in proceedings and book consecutively. She got second prize for oral paper presentation at Nagpur. She has completed 3 projects with different funding agencies and 3 more major projects are going on. Under her guidance, 4 students have already completed PhD and 4 are ongoing. She has also served as a reviewer approx in 9 journals. She has also organized various approx total 10 short-term courses, training courses, national workshops, and international conferences, and also delivered 6 invited lectures. Matter of proud that she owns 2 patents. We all are desperately waiting to hear our resource person, Dr. Radharani. She is going to deliver her talk on topic microbial fuel cell, an emerging technology for bioelectricity generation and simultaneous wastewater treatment. So now I invite you, madam, welcome. Before that, I would like to say all participants, you may rouse your queries and curiosities in chat box. Now I am handing over this floor to Dr. Radharani. Welcome, madam.
Thank you, ma'am. Thank you, Dr. Sushila Srivastav, for such a nice introduction. And first of all, let me congratulate the members of organizing committee for conducting this international webinar on a very relevant topic, emerging trends in biotechnology. And I also extend my warm greetings to Dr. Suresh Selawat, Chief Patron and Additional Director, Department of Higher Education, Indore Division, Dr. Shobha Jain, Patron and Principal of the college, Dr. Rajendra Kochale, Coordinator and Convener of this webinar, Co-Coordinator Dr. Sushila ma'am, and co-conveners Dr. Anita Solanki, Dr. Madan Waskil, and a very dear friend and colleague of mine, Dr. Monica Dhote. So when I was approached by the organizing committee members to deliver this talk, I immediately said yes, because being an academician, I never want to lose any opportunity to interact with students and share my research experience. So first of all, before being a researcher, I am a teacher. I am working as assistant professor at Department of Biotechnology at National Institute of Technology, Allahabad, Prayagraj. Let me share my screen with you people. Is the screen visible?
Yes, yes.
Ma'am, it's visible.
Okay.
Please render it in the presenter's mode, ma'am. Yes, yeah. It's fine, ma'am.
So the topic of today's talk of mine is microbial fuel cells, an emerging technology for bioelectricity generation and simultaneous wastewater treatment. So let us begin with defining the problem a bit. Today's developing world, especially the developing countries and underdeveloped countries, are dealing with two major issues. The first issue is that of availability of pure water, treated water for various domestic applications. And the next major issue is the energy crisis. So this increased urbanization and industrialization has given rise to the water pollution issue. So here we can see in this diagram that there's so much amount of sewage, both domestic and industrial, is generated per day, and a significant percentage of this goes into the environment untreated because we don't have that much sewage treatment capacity. Various industries like tanneries, fertilizers, textiles, refineries, and other food industry, they all generate a huge amount of contaminated effluent water, which is contaminated with both organic and inorganic pollutants. So this is a brief outline of the global scenario that currently we have 66.3 billion people which don't have access to fresh water, and a significant population around the world is compelled to drink contaminated water in absence of availability of pure drinking water. And in our country also, we are aware government is running so many programs like the Clean Ganga Mission and several other programs to purify our rivers, because we every day witness that so many untreated sewage lines, untreated effluent lines which are coming from industry are directly dumped into our rivers, which ultimately give rise to pollution. This is a diagrammatic representation of use of water in different sectors. Like agriculture remains the largest consumer of water, followed by domestic purpose, particularly drinking, followed by industry, energy, and other sectors. Now we have discussed about the first issue, that is availability of pure water in absence of enough mechanisms which are available for treatment of the effluent which is generated from housings as well as industrial sector. The second main issue is that of energy, and due to the shortage of non-renewable energy sources, we have to shift ourselves towards renewable energy sources. And here energy demand is projected, and it is expected to increase by around 85% by the year 2040. And this is the main reason why we need to work on several technologies which can be applied for both the purposes, like any such technology which can be used for treatment of industrial effluent or domestic effluent along with generation of bioelectricity. And some of the traditional methods which are very commonly applied or used in sewage treatment plants include activated sludge processes, trickling filters, aerated lagoons. We all are aware about these things. And we also know that all these techniques, they are energy intensive. They require a high energy input, which limits their field application or their availability throughout the societal system. Now let's come to our core concept, the concept of microbial fuel cells. So before going to microbial fuel cell, let's in brief talk about fuel cell. The concept of fuel cell was first introduced by William Grove in around mid-18th century while he was experimenting with electrolysis of water. And we all are aware that in electrolysis, when electric energy or electricity is applied to water, it breaks down into its individual components, that is hydrogen and oxygen. So he thought of the reverse reaction. He thought if we can combine the two things, that is oxygen and hydrogen, can we get electricity along with water? And he experimented on that, and he found out that yes, it is possible. And that way, the concept of fuel cells was introduced. So a fuel cell is nothing but it is an electrochemical device which is used to produce electricity using some chemical compounds. Say, for example, very basic chemical compounds are hydrogen and oxygen, which can be combined to give water along with electricity generation, just the reverse reaction of electrolysis of water. And many chemical fuel cells have been extensively used even in space missions and various other applications. But now let's come to the core topic. What we are interested in talking about are the microbial fuel cell. Microbial fuel cell, they are bioelectrochemical devices or bioelectrochemical systems. And here, the chemical energy of organic compounds is converted into electrical energy by microbial metabolism. So in a manner, they differ from fuel cell because they don't completely depend on chemical reaction. Rather, they depend on microbial metabolism, or we can say microbially catalyzed metabolic pathway for the generation of electricity. And this concept, the concept of microbial fuel cell, is becoming attractive because it can provide an alternative method for generation of green electricity or clean electricity, and it can be done by using the waste water or other waste material which is generated as a form of industrial or household effluent. And hence we can say that the microbial fuel cells have the potential of treating wastewater with simultaneous electricity generation. So this is a very broad diagrammatic representation of setup of a microbial fuel cells. We need to understand some important parts. So first of all, it is a fuel cell. It is going to generate electricity. So it is divided into two compartments, the anodic compartment and the cathodic compartment. In anodic compartment, the electrode which is present, it is anode, whereas in cathodic compartment, cathode is present. And the two compartments are separated by a semi-permeable membrane which allows only the flow of protons. It don't allow the flow of electrons. It is specific for protons only. And many a times, hence in many literature, you may find the term proton exchange membrane, but it can be anything which is specific for protons only, and it allows the transport of protons only. Now, among the two chambers, the anodic chamber and the cathodic chamber, the anodic chamber is the biological one where our microorganisms exist. They do their metabolism. They secrete electrons as a part of their metabolism. So here the anode is essentially a bioanode. And here the electrolyte can be wastewater. It can be any medium which can support the growth of microorganism. And another very important peculiar feature of anodic metabolism is that this chamber is anaerobic, whereas this chamber, that is the cathodic chamber, has to be aerobic. Even you people can set up, or you people can prepare a small setup of a microbial fuel cell and get some energy output from it. But the very important thing one needs to understand that electrons are released into the extracellular environment under anaerobic metabolic conditions only. So anodic chamber has to be devoid of air, whereas cathodic chamber generally contains air because oxygen here acts as a terminal electron acceptor, and hence it facilitates the cathodic reaction. So here in anodic chamber, as we have seen, the organic matter is present as anolyte. We have our bacterial cells. In general, the bacteria or the microorganisms which have the capability to secrete electrons extracellularly are termed as electroactive bacteria or exoelectrogenic bacteria. So under anaerobic conditions, they metabolize the organic matter which is present in the anolyte and convert it into simpler compounds like CO2, and they produce electrons. Protons, they cross this barrier, the proton exchange membrane, and they move to the cathodic chamber, whereas electrons do not have any other way to go out. When the electron density increases here, the only way by which electrons can move is through the external circuit. And when electrons are forced to travel through the external circuit, we know electricity is produced, because electricity is nothing but it is a flow of electrons. And since in this schematic representation, we have talked about two chambers, so this is a representation of a dual chamber microbial fuel cell. But in various other reactor configurations, we can also have a single chamber fuel cell, microbial fuel cell also. These things I have already talked about: the anodic chamber, which is anaerobic one, the cathodic chamber, then the membrane which separates the anodic and the cathodic chamber, which can be done by having a salt bridge or by connecting the two chambers using the salt bridge, or it also can be done by using specialized membrane. One of them is Nephron membrane. Then we have electrodes, any conductive material can be used as electrodes, but some properties are desirable for electrodes. Some of the very common material like carbon cloth, carbon brush, graphite, some metals like copper, zinc, stainless steel, they are very commonly used as electrodes. But the properties which are required for electrodes, anode and cathode, differ a bit. For example, in anode, one very important property which is desirable is the biocompatibility. If any material which has to be used as anode, it should not be toxic for microorganisms or bacteria, because microorganisms are going to attach to its surface, and this attachment facilitates the flow of electron and hence the efficiency of your microbial fuel cell. So a very first desirable property, which is very important in case of anode electrode, is biocompatibility. Apart from that, anode should also have a good surface area, a large surface area. Again, a good large surface area ensures the attachment of microorganisms, and जितना बड़ा surface area होगा, microorganisms उतनी effectively attach होंगे, and hence the system or the fuel cell will give good results. Then we need some external wires to join the electrode and to get the external circuit and substrate, which is actually the anolyte or the electrolyte, which is present in anodic chamber, which can be wastewater or it can be made from various media composition using substrates or easily utilizable substrates like glucose and acetate. So the concept or the system, it works on redox reaction, and organic matter is oxidized by microorganisms, more specifically bacteria, thus causing the release of electrons in the extracellular environment. And these electrons, they are transferred from the anode to the cathode through an external electric circuit, which leads to generation of electric current. While the protons, they are transferred from anode to the cathode through the proton exchange membrane or the salt bridge, whatever mechanism is being used in that cell. Now, these electrons and protons are consumed in the cathode compartment, where they combine with oxygen to form water. Now here we can see the two reactions which are occurring at the two electrodes, that is anode and cathode. At anode, as we have already discussed, that metabolism of organic compound, here we have taken the example of glucose, under anaerobic conditions happens with the release of simpler compounds, like here in this case it is CO2 along with electrons and protons. Whereas in cathode, in the cathode cell or the cathode half of the cell, the protons and electrons combine together in the presence of water to form water. And the electrons which were produced in the anodic chamber, they travel through the external circuit, and this travel or this flow of electrons give rise to the electric current. Now let's talk about electrodes in brief. Though I have briefly mentioned most of the points, but still, once again, let us discuss about them. The physical and chemical properties of electrodes are important because they affect the microbial community, the microbial diversity, and especially in case of anode, this is important because it is the site where microbial metabolism happens. And hence, as we have already discussed, that an anode should be biocompatible and it should have good surface area apart from being electrically conductive. So electrodes with high surface area, conductivity, and biocompatibility facilitates the attachment of microbes, and hence gives better results in terms of power output. And also it significantly affects the overall economics of the process because they add to the overall cost of the setup or the overall cost of the reactor. Now, this is an image of some of the materials which are these days commonly used in microbial fuel cell reactors, like we have carbon cloth, carbon mesh. These are brush which are made of carbon fibers. Then this is graphite, various forms of graphite, modified forms, unmodified forms, and also metallic electrodes are also used like stainless steel. Now let's talk about biocatalysts. So here microorganisms are the biocatalyst. In reference of a microbial fuel cell, whenever we use the term biocatalyst, it essentially means the microorganisms which are actively present there. So these are basically exoelectrogenic bacteria or the electroactive bacteria, which are the main functioning things there, or which are the main soul of the whole setup. And some of the very common, well-known exoelectrogens include Geobacter sulfur-reducens, Alkaligen faecalis, several species of Pseudomonas, then Aeromonas hydrophila, Shevanella putrefaciens, then several bacteria belonging to the family of Proteobacteriaceae, and several Enterococcus. So here, one very specific and important thing to keep in mind is that these microorganisms are either facultative in nature or they are strict anaerobes. Because as we have already talked about the fact that generation of electrons in an extracellular environment can only happen under anaerobic metabolic conditions only. So whatever microorganisms we are talking about, including these and many other electroactive bacteria, they all are either strict anaerobes or facultative microorganisms. And facultative microorganisms are those which can metabolize under aerobic as well as anaerobic conditions. Now let us have a brief look at the electron transfer mechanism, how electrons are transferred from the bacterial cell or the microbial cell to the anodic surface. So there can be a direct electron transfer mechanism, or there can be mediated transfer mechanism as well. It depends again on our setup. Many of the microorganisms, they have the capability to direct transfer their electrons to anode. They don't require any additional mediator to be added in the medium or to be present in the anolyte. And this is done through several features or certain special features with which they are equipped. Like some of them have outer cytochromes which act as electron carriers, and they pick up electrons from microorganisms and directly transfer them to the anodic surface, while others have special pili-like extensions, and you may find the name nanowire in many literature, and you can also visualize them. They are commonly termed as nanowires to mimic or to understand what kind of function they are performing, because they act as nanowire in terms they conduct electrons, and in this manner the electrons are transferred from one microorganism to the other, or from one surface to the other. Like in this case, the electrons, they are transferred from the microbial cell to the anodic surface through nanowires. So these are the direct mechanism. Direct mechanism means microbes have their own provision for transferring electrons. They do not require any additional mediator or any additional electron carrier to be added in the anodic chamber. Whereas in mediated electron transfer mechanism, several mediators are present, which most of the times we have to add or we have to supplement in the anolyte. And these mediators are nothing but they are certain compounds which can exist in the redox form, in various redox forms, either as reduced forms or the oxidized forms. So they act as electron carriers. They carry the electron from microorganism and they transfer electrons to the anodic surface, and then again they are oxidized, and again they are ready to take the electron. So microbial fuel cells, apart from being a device which can be used for generation of green electricity, they have very wide application. They can be used, and in fact, they are extensively used for wastewater treatment as well. And the range of wastewater for which they can be used is again diverse, because here the catalyst which we are using, which are the microorganisms, they have tremendously immense catabolic potential. So depending on the type of microorganism you are using, depending on the type of exoelectrogens we are using, we can target any type of waste we want to. Like it can be municipal wastewater, which largely is the sewage, domestic sewage, and it is rich in BOD and COD, nitrogen and phosphates. Or it can be waste from farms. If it is wastewater from agricultural fields or agricultural industry, then it contains several organic pollutants like pesticides and fertilizers. And wastewater coming from other industry is rich in other recalcitrant compounds or other pollutants, like we have polyaromatic hydrocarbons, we have dyes which come from textile industry, we have metals which come from electroplating industry. And in most of the setup, it is found that from 50 to 90% of the organic solids, they are degraded, they are consumed, they are metabolized by microorganisms. Or we can say they are degraded by the microorganisms. And a COD removal of up to 90% is achieved or can be achieved using a microbial fuel cell. And in terms of coulombic efficiency, it can yield up to 80% coulombic efficiency. Yet another subtle application of microbial fuel cell is that it can be used as a biosensor itself, because the current which is generated from microbial fuel cell is directly proportional to the BOD levels of, or the strength of organic matter which is present in wastewater, and hence that way slight modifications in a microbial fuel cell can give it a feature where it can be used as a biosensor to measure the strength or to measure the BOD, COD levels of any given wastewater sample. Yet another important feature, interesting feature rather, of microbial fuel cell is its application for generation of biohydrogen. So with slight modifications, microbial fuel cells can be used for generation of biohydrogen as well. So irrespective of the fact that this technology has so much advantages and such a wide application, still it is not yet being exploited on a large scale at field level because of its few limitations. A very important limitation is generation of a low power density, and this low power density is largely because of certain losses in the current which are due to internal resistance which is provided by the electrolytic solutions, like the activation losses or the ohmic losses, and many times bacterial metabolic losses. And the second feature, the first disadvantage which we have talked is low power density, and it may be due to the activation losses, ohmic losses, or metabolic losses. And the second is the initial cost of the setup is relatively high. Now, we have talked about a general overview of the technology, of the setup, and how it can be applied for various aspects, like for wastewater treatment as a biosensor, for hydrogen generation, for bioelectricity generation. Now I will be talking in very brief about some of the work which we are doing. So we have worked on different type of reactors. We have worked on very miniaturized reactors where the reactor volume was around 800 microliters. And starting from that, we have worked on large reactors as large as the volume capacity of few liters, 3 to 5 liters. So today I'll be giving you an overview of the large reactor which we are working on, and it is a multi-modular reactor. So multi-modular essentially means multi-electrode. So exactly, it does not have number of cells. We can say it has number of modules, and each module act as individual cell, and many modules, multiple number of modules can be attached to this kind of setup or this kind of reactor to scale it up. So the beauty of the reactor is that it can be easily scaled up. So this is the overview of the reactor, and this is each module. Like if we start from here, 1, 2, 3, 4, 5, 6. So this is a 6 modular setup, and each module, as I said, behaves as an individual cell. So we have individual electrodes in each module. This, the outer electrode, it is cathode, and here it is exposed to air. So it is, in such setup, it is known as air cathode. And this is the anodic chamber where the wastewater or the analyte is present. And here anode is present. Anode is dipped in this chamber. And all these electrodes, they are connected through an external circuit using someone is scribbling my screen. Please don't do it. Please don't scribble the screen. Okay, so let us briefly discuss operation. So actually, a simulated wastewater was created by adding some of the chemical compounds, and the setup was run. The inoculum used was a mixed culture inoculum, which we have enriched from various habitats, and it was operated in batch mode. So we studied basically 2 parameters. The very important parameter was of electrochemical properties. So we studied the electrochemical properties, which included operating voltage and open circuit voltage. So open circuit voltage is that voltage which is without any external resistance, whereas operating voltage is calculated when we applied an appropriate external resistance. So both the voltage were collected and using a digital data logger system, and power density and current density were calculated using Ohm's law. So this is the setup. This is the reactor which is being operated. This is the data logger system, and we get a continuous input of data, how the current and voltage and resistance of the reactor or of the system are changing. Let me present you with some of the findings. So these initial two graphs are of the generated voltage, where we have the open circuit voltage and operating voltage. As I have already told you, open circuit voltage is the voltage which is measured across the open circuit. That means we have not applied any resistance to it, whereas operating voltage is the voltage which is measured by applying resistance. So it is measured across resistance. So here we can see it produced maximum voltage at around 15 days, and thereafter there was a kind of stationary phase which later on slightly declines. This was because it was operated in batch mode. If operated in continuous mode, when the nutrient supply is continuous or the wastewater input is continuous, we can expect this phase to be longer. And this is the power density and current density graph. So whenever we express the output in terms of electricity generation, it is expressed as power density, where power is expressed as watts and power density as watts per meter square. And this meter square is the dimension of the electrode which is used. Yet another interesting study which we carried out, as I have talked also about it, was the enrichment of exoelectrogenic bacteria. And we have tried to enrich exoelectrogenic bacteria from various habitats. And some of our habitats, common habitats, included cow dung, then anaerobic sludge, marshy areas. So we used to collect the sample, then we measure it, and then we keep it for enrichment. And then this inoculum, it was used as a starter culture to operate a small scale reactor. In this manner, electrochemical enrichment was performed. And here we have given the comparative results of what were our observation in terms of voltage output using two different samples, the cow dung sample and the domestic wastewater sample. And whatever results, somewhat results I have presented with you regarding the multimodular design, it's working. And our exoelectrogenic bacterial inoculum, which we have enriched from cow dung. So on basis of these findings, an patent has been filed. This is yet another method of enrichment of exoelectrogenic microorganisms. So here we put or we inoculate our sample, which in this case can be cow dung or anaerobic sludge or wastewater sample or a marshy sample, into a special medium which contains various salt along with ferric chloride. And then this has to be kept in a cerium bottle, and then we sparge nitrogen to it so as to maintain anaerobic conditions, so that strict anaerobes are also allowed to grow. And then over a period of time, the color of the medium changes. This is because of conversion of Fe3 to Fe2 because of the release of electrons in the exterior environment by the microorganism. So this color change, it is an indicative that yes, exoelectrogenic microorganisms have been enriched, they are growing. And later on, once we have used our inoculum for running or for operating our microbial fuel cell reactor, then again we study the microbial diversity of bioanode, because that is very important. Most of the important exoelectrogenic microorganisms, they get stick to anode, where they deliver electrons or where they release electrons. So we generally, once the reactor has been run for its complete run time, if it is in batch mode, we take out anode and we perform various studies, like we perform microbial diversity studies, we perform the strict culture method, cultural diversity, and maybe some kind of fluorescent microscopy or scanning electron microscopy, just to understand the attachment of microorganisms. So that particular anode is washed off using phosphate buffer, and the exoelectrogenic microbes, they are allowed to enrich in a particular enrichment media. And then we have given our samples for NGS analysis for determining or for studying the non-culturable bacterial diversity of the exoelectrogenic microorganisms which are present therein. But we can also perform the culturable diversity by just streaking our culture on Petri plates. And we have done so, and we have isolated few important and interesting microorganisms here. And here in this table, some of the results of morphological characteristics of particularly the colony morphology are presented here. And here other simple morphology results are presented here. These are very basic techniques which any biotechnologist or a microbiologist use to characterize its microbial or a bacterial strain. Like we have gram staining, which differentiates bacteria on the basis of its cell wall structure into gram positive and gram negative. We have capsule staining, which allows us to identify whether the microorganism or the bacteria is producing capsule or not. And then we have endospore staining, which help us to understand whether the microorganism is endospore forming or not. Then we perform some biochemical characterization to study the biochemical properties of microorganisms, which we have isolated from our anode in detail. And then we got 4 isolates, but out of these 4 isolates, we have extensively studied one only. And later on, it was further studied, it was further identified on the basis of 16S rRNA sequencing, and it was identified as Pseudomonas gesarti. And this is a very beautiful or a very capable bacteria, because it has very a good amount of catabolic potential, being capable of catalyzing the degradation of many organic compounds, and also it is an electroactive bacteria. So from our setup, which we have discussed, of the 6 modular microbial fuel cell, the power density and current density that was obtained was 911 milliwatts per meter square. This is power density, and 1687 milliamperes per meter square, the current density. And COD removal was found to be approximately 67% during 45 days of continuous MFC operation in batch mode. And Coulombic efficiency of the reactor was found to be 0.96%. So this was a brief outline of what work we have performed, what are the findings which we have observed. Now let's come to the conclusion. What are the conclusions which we can draw from our experimental findings? So we have isolated and identified several exoelectrogenic bacteria from different habitats, and one of them was identified as Pseudomonas gesarti on the basis of 16S rRNA sequencing. And Pseudomonas gesarti again was used as an inoculum in various sets of microbial fuel cell, and it was found to be an efficient electroactive bacteria because it gave us good results, even if when it is inoculated as a single bacterium. And hence 16S rRNA gene sequencing was performed, and the sequence has been submitted in NCBI with accession number this. The power density and current density which was obtained, I have already talked about. The COD removal and Coulombic efficiency, I have already discussed about it. So we obtained a COD removal of around 67%, and Coulombic efficiency of approximately 0.96%. One very unique feature while working on MFC is that if we are simultaneously targeting 2 problems while working with MFC. The first is generation of electricity, or rather we can say bioelectricity, and the second one is wastewater treatment. So these 2 properties somewhat are inversely proportional. If in a reactor we will focus or we will optimize, we'll try to get maximum power output, in that case we won't be able to get a good COD removal. And conversely, in the same fashion, if we are targeting towards getting a good COD removal, or maybe we are targeting the removal or degradation of some very recalcitrant pollutants, in that case, we won't be able to get good power output. So it is yet a challenge to optimize the system or to optimize the reactor so that we can address the 2 problems at least to somewhat satisfactory level, if not at the best. And these days there are several pilot scale plants which have been implemented in field, and they have been integrated with traditional sewage treatment plants, and they are working, they are functioning well. So with these small steps, I think we are moving ahead towards the practical application of this technology to the field. And that is what we in our lab are also trying. So we are also trying, and we are also working on scale up of our reactor, where we are developing a pilot scale reactor, and where we will directly, assessing the technology at the field level, where direct input is coming from the domestic when it is directly going into a sewage treatment plant, which we have designed, and there we have integrated a multi-modular microbial fuel cell after primary clarifier tank. So it is a kind of replacement of secondary treatment methods like activated sludge. So this was all about the concept, the technology, and some of the results, which or some of our findings. And these are some of the publications related to microbial fuel cells. So we have also worked on miniaturized system, as we have talked about. So it has been published in Bioresource Technology. Then we have also worked on modification of electrodes so as to increase the output, the power output, and some of the findings have been published. Then we have also tried to understand the microbial community dynamics which happen or which occur in a microbial fuel cell reactor, and some of our findings have been published here in this paper. And we have also filed 2 patents. One is on the novel composition, which is used for surface modification for increasing the power output, and other is on obviously our design, which we have already, which I have mentioned in brief during my discussion. So I would first of all like to express my sincere gratitude to Science and Engineering Research Board, which have funded our project. The findings of the project or the work which I have shared with you, it is the outcome of this SRB funded project only. Then I would also like to thank MNIT Allahabad, and a special mention to my PhD students. Dr. Gitanjali has completed her thesis and she worked on anode modification. Currently she has joined, I think, IIT Roorkee as a PDF. And Roma is still continuing her PhD on microbial fuel system. So with this, I conclude my talk. I conclude my presentation. Thank you for giving me your time and attention. And I'll be happy to have some queries.
इसको बंद कर थोड़ी देर में.
Hello. Hello, yes.
Thank you so much, Dr. Radha. It was such an excellent talk and sharing your practical knowledge with us. We are so glad to hear you.
मैम वॉइस अनम्यूट कर लीजिए आपका.
Dr. Monica, you are not audible.
Monica, madam, please unmute yourself.
Okay. I'm sorry, everyone. Thank you, Dr. so much, Dr. Radha.
Monica, madam, please unmute yourself.
Okay. Ma'am, is it audible? Dr. Radha, is it audible?
Yes. Now you are audible. Now you are audible.
Okay. Thanks. I'm sorry. Thank you so much, Dr. Radha, for such an excellent talk and sharing your practical knowledge and practical experience with us. It was really wonderful.
Pleasure is all mine. Pleasure is all mine. I am anytime ready to share my experiences with students. I myself am a teacher, and it's really an opportunity for me to share my experiences with young generation of ours.
We hope and we wish you will deliver us excellent treatment technology for wastewater and NFCs to society and scientific society, and yeah, we will see you in top position. Okay, and for that, I request Dr. Madan Master sir to start question and answer session. Okay, now sir, it's over to you. Thank you, Radha.
Vaskil sir, please unmute yourself.
Thanks for Dr. Monica Madam for invitation me. I, Dr. Madan Singh Vaskil, Faculty of Zoology, Government PG College, Mahu, to ask the question from Dr. Radha Rani, the second session speaker. The questions are as follows. What are its field applications?
Unmute yourself.
Me unmute. Okay. Thank you for the interesting question, because even as researchers, if we work on any technology, we have to look for its feasibility in field applications. So that is a very important aspect for any researcher. So as far as the field application is concerned, microbial fuel cells are well applied in field at pilot scale. अभी बहुत लार्ज स्केल पे हमको नहीं मिलेगा, जैसे कि MLD लीटर्स के स्केल पे, जैसा कि नॉर्मल सीवेज ट्रीटमेंट प्लांट्स में होता है, बट पायलट स्केल प्लांट्स आर अवेलेबल एंड दे हैव बीन इंटीग्रेटेड विथ मेनी इफ्लुएंट ट्रीटमेंट प्लांट्स एंड सीवेज ट्रीटमेंट प्लांट्स, एंड the technology has application especially in rural areas or the areas where getting a continuous electrical supply is difficult if not impossible. तो ऐसे इलाकों के लिए if we can have a pilot scale setup, it can be used, it can be of emergency use in such setups. And of course, for other industrial application, for household applications, pilot plants are available and they are successfully running.
Thank you. Second question, ma'am. What about cost, economic value?
Yes, it is yet again a very important aspect. So initial cost of implementing or initial cost of construction is the main consideration, but once the reactor has been set up, once the fuel cell has been set up, then it is not a big problem. It can be maintained because we are also harvesting energy from it. So that is a kind of output which we are getting. So agar hum in dono ko energy balance kare, then it is economically viable.
Okay, last question, ma'am. Environmental change on microbiome.
Environmental change on
Microbiome.
microbiome.
जी.
Microbiology.
Microbiome.
Microbiome.
जी.
Okay. See, environmental change and climate change, it itself is a big issue, and itself is a big topic of discussion. And obviously, apart from affecting various biotic components of the ecosystem, the flora and the fauna, it is also affecting microbial strains. It is also affecting the microbial community across the globe. So obviously, it is affecting in a bad way. We are losing our diversity with respect to microorganisms of any habitat.
Thank you, madam. Now, I would like to invite Dr. Anita Solanki for word of thanks.
Thank you, sir. Am I audible?
Yes, you are audible, ma'am.
Am I audible? Audible?
Yes,
Yes, you are audible. You are audible.
Thank you, Vaskil sir. Good evening to all of you. On behalf of the Department of Biotech, I would like to propose my vote of thanks to each and every person who has contributed significantly to the success of this international webinar. First of all, I would like to give special and hearty thanks to Dr. Priya Nirgaonkar and Dr. Radharani ma'am, who delivered most useful and highly informative lecture today. We are thankful to Honorable Dr. Suresh T. Silawat, sir, Additional Director of Indore Division and Principal Government Holkar Science College, Indore, who gave us his valuable time and blessings. We are thankful to External Advisory Committee members, Honorable Dr. N.K. Dhakar, ex-Vice Chancellor, DDBE Indore, India, Dr. Asha Jwarkar, Dr. Abhay Fulke, Dr. Anil Kumar, Dr. MMP Srivastav, Dr. Shweta Kalve Kulkarni. From them, we have been receiving high quality advice for this webinar. We are especially thankful to our principal, respected Dr. Shobha Jain, Madam, who always support to every creative work. Last but not least, we are thankful to College Advisory Committee member and Dr. Swagata Gupta, Dr. Rekha Verma, and all faculty members, student representatives, media and communication committee members, technical and other supporters of making this phenomenal webinar as a grand support. Thank you to Dr. Rajendra Kochle and entire team members of the organizing committee. Congratulations, all of you. Once again, thank you so much, everyone. Thank you.
Thanks a lot. The biotechnology department, it was an exemplary international webinar with two very quality-driven themes and topics. And with this, we close today's webinar. Shekhar Bhaiya, please close kar dijiye.